Today the topic of my lecture is biofuel effectively and easily by microbes from carbon dioxide and water. So first we have to learn what is actually biofuel, and to reveal what that actually is we have to go through a few definitions. And first is the biological carbon fixation. So carbon can be inorganic or then it can be organic, and organic is from living material. So inorganic carbon in form of carbon dioxide, so that can in nature be used for making organic matter. So the practical example is when a plant takes up carbon dioxide and uses solar energy and produces then oxygen and sugar for growth. So that's how this CO2 is fixed by a living organism, and that's carbon fixation. Then you have biomass. And what is actually biomass? So that's the biological material that is used for biofuel production. So the example of that kind of material is, for instance, kernels of corn or mats of algae, and then it can be forest residues like small trees or stumps or branches from the forest. So then we can start producing biofuel, but how is that formed? So that is actually produced by or from a living organism, and it's produced from overmentioned biomass, and it's produced in a short or relatively short period of time. So this biofuel can be any hydrocarbon fuel that is produced from organic matter. So when we talk about biofuels, so then we have to also think about what is renewable energy. So since biofuel is produced from living organic matter, it has to be harvested. And the biomass is renewable because it can be grown whenever needed in a relatively short period of time. And that's in opposite to fossil fuels that are also organic matter that readily can be used for energy, but that has been formed during millions of years of development. So there are different kinds of biofuels, and the liquid biofuels are the most common ones, and that's ethanol, biodiesel, methanol, and biobutanol. So again, example is like in Brazil, the gasoline contains up to 95% of ethanol. But in other countries or in Scandinavian countries, it's mostly 10 to 15% of the gasoline that is replaced by ethanol. Another example of biofuel is biodiesel. And that's again a fuel that's composed of so-called monoalkyl esters of long-chain fatty acids. And that you get from vegetable oil or animal oil. So these biofuels can also be gases, and they are produced in a biotechnological manner. So apart from these ethanols and vegetable oils and animal fats, so gases can be used for energy production. And a practical example is then, for instance, plant and agricultural waste that can be put into bioreactors, and in the bioreactor then the gas is produced, and in this case when you have plant material and animal material you can produce methane. And methane is a greenhouse gas. But in this biotechnological process, of course you need the microbes, because it's both bacteria and archaea that produce the gas from this biomass. And they produce it in these bioreactors without oxygen, so the environment is anaerobic. So having said that methane is a greenhouse gas, so then we see that there is a connection to climate change. And climate change is generally thought that it's caused also partly by human action, and the significant increase of greenhouse gases has been recorded in the atmosphere. And actually the concentration of carbon dioxide, which is a greenhouse gas, so that has increased by 40%, then methane concentration in the atmosphere with 150%, and the concentration of nitrous oxide has increased with 20% since the time before our industrialization started. So here on the picture to the right you can see here is the CO2. It's on the x-axis you see the time scale, and on the y-axis you see the concentration of the gas, and you can see that this curve is going up for CO2, especially here in the millennium, around the millennium, and the same goes for methane. The curve is going up, and also for nitrous oxide. And here we have a bit the same thing. We have the time scale here, and then we have the different greenhouse gases and how big part the emissions are between 1970 and 2010. And we can see here in dark blue we have the nitrous oxide. Then in light blue here you see the methane, and then in the dark brown, so that's the carbon dioxide, and the light brown is then carbon dioxide from fossil fuels. But again, if you look during these last 40 years, you see that the curves are going all the time up. So the microbes are important in climate change, both as producers and then consumers of these greenhouse gases. So now a little biological background. So the microbes, they have a central role in the living environment and the whole biosphere in nutrient cycling. So if we take the nutrient carbon and nitrogen, so they regulate both these nutrients in both in the atmosphere and also in the soil or in the water. And in these microbial processes, these greenhouse gases are both released and then fixed. So they are most produced but also used. And microbes, so the archaea are producing, for instance, methane, but then there are bacteria that can consume again methane. And then due to the large spread of microbes on Earth, so these microbes really have a big role in affecting the climate. So methane, where can we find, or where is methane naturally produced? So that's in wetlands, and mires or peatlands are the source for methane, but some peatlands could also function as sinks. So that means that there is no methane emitted from the peatland. But that depends on what type of peatland and what the water level is and the nutrient situation is. And it's the methanogens there in the peatlands who produce the methane, and they belong to the phylum Yrjälä. So then we come to the more practical question. Methane, is that a threat or maybe a chance? We have just seen that it's a greenhouse gas, but we could also produce energy from it. So methane is formed when this organic matter is degraded, and it's produced without oxygen anaerobically, and it's at the end it's the archaea who make the methane, but before that the bacteria have been breaking down the organic compounds. And we find these archaea naturally in these mires, but we can also find them in rice fields, and actually we can find them in human-made landfills. In most places where there is no oxygen, but there is biological material or organic material to break down. But thirdly, these methanogens, they are also in rumens of animals, and also they can be found in sediments wherever oxygen is depleted. So from carbon dioxide, like we see here, carbon dioxide and hydrogen, this methane is produced, and then we also get water. So now I will tell you about the chance we have. So I told you that methane can be used as fuel, and actually it can be used in the form of natural gas or biogas. And these microbes, methanogens, together with bacteria, can be used for producing this methane. And now in this technological invention, the idea is to mitigate climate change by utilizing this process called hydrogenotrophic methanogenesis. So now we will have a look what this technology looks like. So first, these methanogens, actually they have not been too much studied, so it was only in the beginning of the millennium that they were first detected from DNA samples in our mires, and there are doctoral theses that have been done at the University of Helsinki regarding these methanogens. So what's the practical way to produce energy, in this case now, in this invention, from methane? And this example now comes from the Natural Resources Institute, LUKE, where they have developed a very peculiar bioreactor. So what they did, first they went to the mire or the peatland, and they took samples from about 2 meters depth to get a microbial consortium, and then they were working a lot with that in that microbial consortium to start producing methane. And then when this consortium was somehow ready, they put it in a bioreactor. And this bioreactor is a fixed bed bioreactor, So it means that there is no biomass except the microbes in this bioreactor. So there's no biomass needed. And the starting material is therefore gases, and what you get out from it is water and then the methane gas. So this technology is using renewable energy sources to produce synthetic natural gas from CO2 and hydrogen. And what's especially good with this invention is that you can transport renewable energy. So let's say you have wind power or solar energy locally, and you have a lot of wind in the middle of summer and a lot of sun, and you get a lot of electricity, but there is no use for it. And electricity is difficult to store, so what you then could do, you could transform this electrical or wind power energy, you could transform it to methane. You could produce methane. And this methane again could be utilized in existing infrastructure in society. So let's have a look at this picture, which tries to explain what the fine idea with this bioreactor is. So if you have in a farm, or it's an industry or something that is producing a lot of CO2, so that CO2 gas can be channeled into this bioreactor. And then again, if you have electricity, so what that could also come from, yes, from this solar energy. So then you can use that to break down water, and then you get hydrogen for this bioreactor. And then in a certain amount of time, the methane is produced, and in this case, you could, when it's clean methane, you can use it for fuel in cars. But of course, you could also use it for heating if you want. But the idea is that you have a bioreactor where you don't have biomass, but you introduce hydrogen and CO2 to this bioreactor. So you introduce gases instead of biomass. So you actually can, with this invention, you can bind emissions and carbon neutral products. So the crucial benefit is really that this device can capture emitted CO2 from different production sources. And then this is a very flexible bioreactor, because you can freely choose where you take your hydrogen or carbon dioxide from. And then apart from that, with this bioreactor you produce methane, it can also be used for microbiological protein production and also the chemical industry. So there's a lot of different uses for the produced methane. So still another picture here to tell the very diverse use of this bioreactor. So we get in carbon dioxide, and it can come from all these different sources. Then you need the hydrogen, and it can come from solar energy, wind energy, or other renewable sources, but also from synthetic gas. But all this means that you have to have these sources close around, or actually you can install your bioreactor close to these sources of these needed starting materials. And then you get out this methane, and apart from fuel for cars, you can also use it for protein production or other chemical production. So the question for us was also that okay, the starting material for this bioreactor was a sample from a mire, but what is actually growing there in this bioreactor? So we decided, or we got the opportunity to find out who is actually living there in this bioreactor and who is maybe producing the methane in the bioreactor. And this we did using next generation sequencing to study the diversity of the microbes in this bioreactor. And with this technique you are producing a lot of sequences, and every sequence comes from one microbe. And then you get a lot of data, and the challenge will then be how to handle this data, and then you need bioinformatics and biostatistics. So here just what we found. The methanogens in the bioreactor were of several different types, and here they are just mentioned. And the way you can identify these sequences is that you use a sequence database and you look for similar sequences in the database, and through that you are mostly able to identify these microbes in the bioreactor. And we were following this for a few months, and we took 10 samples during 2 months, and here you can see to the left how we have compared the different samples. So every BR is one timely sample, so 1 is the first sample in the beginning of the process and 10 is from the end of the process. And then we have compared these bacterial communities, so with this kind of a phylogenetic dendrogram. And then to the right we did the same with the methanogens that are there in the bioreactor. And the difference is that the bacteria or the bacterial communities have developed in a somewhat linear manner, but then the methanogens have developed in a very different way or different fashion. And from the amount of sequences we could clearly see that there were these 2 types of methanogens that were the most common ones, and probably those that really are those that produce most of methane. So one of them was this methanobacterium, and when we look from the literature what this methanobacterium is known for, so it grows best in 30 degrees, and it has also been detected in rumen of cows. So here you can see that the microbes are all over, but they are performing the same kind of function. And then the other most abundant archaea was this Methanocorpusculum, and this type of methanogen has before been found in processes where methane is produced, and it has been found in wastewater treatment and landfills. But of course here it's only a question of that we have sequences. We have not isolated the microbes, because isolating methanogens is a very difficult process, and that can take half a year or 1 year or 2 years, and it's very difficult. So this is just on the basis of sequences. And when you have these different sequences, you can also look how they are related to each other, so you can make this kind of a phylogenetic tree. And there you see the names of, or the closest neighbors, meaning the best detection that we can give for these sequences that we isolated from the DNA from the water that comes out from this bioreactor. And to conclude, so I can only say that cooperation is smart, because in this project the research findings that we have accomplished so can be used for improving the bioreactor and the bioreactor process. And when we learn about these methanogens, we can also maybe understand more about the major ecosystems from where these methanogens are that are used here in the bioreactor. So this was a cooperation between the Natural Resources Institute and then the MEM group in the Department of Biosciences at the University of Helsinki. So thank you, and I hope you learned something about biofuels, something new.